Hi everyone, welcome to Agri Clarity by Clarity for Sure, your one-stop platform to simplify complex agricultural concepts with clarity and precision. Today, we are diving into a topic that is transforming modern farming, which is genetically modified crops. But why is this important for us to learn? Because India is an agricultural based country and our farmers are facing major challenges like low productivity, pest attacks, and unpredictable climate conditions. The government is promoting GM crops to support farmers in overcoming these problems and ensuring food security for our growing population. As future agricultural experts, understanding this concept will not only help us in cracking exams like IBS AFO, NABARD, and other agri-related exams, but also enable us to contribute towards sustainable and innovative farming solutions. In today's session, we'll be covering the following contents. First, what are GM crops? Second, examples of few important GM crops in India. Next, how are GM crops made? Why is government focusing on GM crops? And ending the session with a meaningful conclusion. First, what are GM crops? Genetically modified crops or GM crops are plants whose DNA has been altered using biotechnology techniques to introduce desirable traits like pest resistance, herbicide tolerance, and higher yields. This means scientists make small changes in the plant genes to improve its quality. For example, adding a special gene from a bacteria called Bacillus thuringiensis, which is Bt, scientists created a Bt cotton, which naturally kills harmful pests like bollworms. This reduces the need for chemical pesticide and increases the cotton production. In our country, there is a well-established regulatory framework for approval of genetically modified crops as per rules for manufacture, use, import, export, and storage of hazardous microorganisms, genetically engineered organisms or cells, 1989 under the Environment Protection Act 1986. The next content of this discussion is examples of GM crops. To discuss the examples of GM crops better, we have divided the examples into three subheads. First, the genetic modification done for making these GM crops, the purpose behind the genetic modification, and the current status of the particular GM crop in India. India has approved a few GM crops for commercial cultivation. Let's look at some key examples. First, Bt cotton. Bt cotton is modified with Bacillus thuringiensis Bt genes to fight bollworms, leading to higher cotton yield. It has been approved since 2002. Bt cotton alone covers more than 90% of GM crops production in India. Next is Bt brinjal. Bt brinjal was developed to resist fruit and shoot borers, though it faced public and regulatory debates. Its adoption has been limited. Next is Bt corn. Bt corn contains Bt genes to protect against corn borers, though its cultivation in India is very limited. Next is herbicide tolerant cotton. Herbicide tolerant was modified to survive herbicide application, particularly developing glyphosate resistance, which is effective for weed management, making weed management easier. Next is DMH11, which is Dhara mustard hybrid 11, is a high yielding mustard variety that is resistant to pest and disease. Recently, this variety has been given limited approval from Supreme Court, making the topic GM crops important from examination point of view. These crops help farmers increase their income, reduce losses, and use fewer chemicals, which is good for environment. Now, talking about Bt cotton, Maharashtra holds first position in terms of area and Gujarat holds first position in terms of production and yield. The top 5 GM crops growing countries are USA, Brazil, Argentina, India and Canada. The crop grown in these countries are soybean, maize, cotton and canola. The next content of today's discussion is the process of genetic modification. Now let's understand the process of making GM crops, which happens in simple 7 steps. First step is identification, which calls for target trait identification. In this, scientists first decide what improvement is needed or what desirable traits they want to incorporate or they want to enhance, like pest resistance, herbicide tolerance, or improved yield. Next step is isolation, which calls for gene isolation. Here, the desired gene is taken from bacteria, viruses, or plant. For example, the Bt gene from soil bacterium is used in Bt cotton to kill pests. Next is modification, which calls for gene modification. Here, using advanced tools like CRISPR-Cas9, which is a DNA sequence of prokaryotic organisms such as bacteria, which is used by scientists either to add or remove or modify the gene to make the plant better. Next step is transformation. In the transformation, there is insertion of gene into the plants. This insertion is done through two methods. First is agrobacterium mediated transformation method, and second is gene gun method, which is also called by particle bombardment. In agrobacterium mediated transformation, a bacterium called agrobacterium is used to carry the modified gene into the plant. In the gene gun method, small metal particles coated with modified gene are shot into the plant cells. The next step is regeneration, which calls for tissue culture and plant regeneration. Here, the culturing of transformed cells into whole plants using nutrients and hormones are done. The second last step is selection and screening, in which using markers of desirable characteristics like herbicide resistance is used to identify the successfully modified plants. Lastly, testing and approval, which calls for field testing and regulatory approval. Here, the new GM crop is tested in fields for safety, performance, and environmental impact before getting approval for commercial use or commercialization. Here is the diagrammatic representation of genetic modification. Let's use the insertion of Bacillus thuringiensis gene in corn as an example. In order to cut out a gene of interest in the bacteria, its total DNA is isolated. Special enzymes called restriction endonucleases act as scissors to cut out the desirable gene. Once the gene is cut out, scientists must make an expression cascade. This consists of additional DNA surrounding the gene so that the corn cell knows where the gene of interest begins and the end. The part that tells the corn cell where the gene begins is called promoter and the end is the terminator. Once the expression cascade has been made, it is inserted into the plasmid. The plasmid is a parasitic cycle of DNA present in bacteria. By putting the cascade into a plasmid, millions of copies can be made. These copies are then introduced into host cell and get inserted into genome. The cells which successfully incorporated the foreign gene into their genome are then expanded in cell culture and used to generate new plants. In this way, the process of gene modification completes. The next content of today's session is why the government is focusing on GM crops. The Indian government is supporting GM crops because they address some of the biggest challenges in the agriculture. First, increased crop yield. GM crops produce more output per acre. Second, reduce pest damage or pest resistance or developing pest resistance. Crops like Bt cotton naturally fight harmful insects, reducing the need for chemical pesticides. Next is lower chemical use or weed management. Herbicide tolerant cotton allows easy removal of weeds without harming the crop, leading to lower chemical use. Next is food security. In food security, with a growing population, we need more food production to feed everyone. Lastly, climate resilience. In climate resilience, GM crops can survive droughts and extreme weather conditions. In simple terms, GM crops help farmers earn more and reduce crop losses while protecting the environment. That's why the government is promoting research and development in this area. Despite these benefits, there are concerns about environmental impact and biodiversity loss. That's why strict regulations and safety trials are conducted before approving any GM crop for use. Here is the hierarchy of key regulatory bodies and their roles involved in successful implementation of GM crops. First is Ministry of Environment, Forest, Climate and Climate Change. They are the overall policy maker for GM crops regulation and ensures environmental safety before approval. Next is Genetic Engineering Appraisal Committee (GEAC), who reviews safety data of GM crops and grants approval or rejection for environmental release. Next is Review Committee on Genetic Manipulation (RCGM), who monitors GM crop research at laboratory and field levels and ensures biosafety compliance. Next is Department of Biotechnology (DBT), who provides funding and infrastructure support for GM research and also facilitates safe and efficient biotechnology advancement. The next is Indian Council of Agriculture Research (ICR), who conducts trials for assessing the performance of GM crops and supports farmers and scientists with technical expertise. Here is the news cutout of recent approval of DMH11. You can read the details of DMH11 and its property, who developed it from here. In conclusion, GM crops are a powerful solution to improve agricultural productivity and support farmers in India. With growing population and increasing demand for food, we cannot rely solely on traditional farming methods. By learning about GM crops, we can support sustainable agriculture, help farmers improve their income, and ensure food security for future generations. Thank you. I hope you found this session informative. For more agricultural discussions, stay tuned to Agri Clarity, where we make agricultural concepts simple, clear, and exam oriented. Subscribe to Agri Clarity for more agricultural insights.